So hello everyone, welcome to tonight's session. I'm Peter Gibbs, I'm a laboratory head at WeHi and a medical oncologist at Western Health. We've got over 100 registrants to tonight's session, which is fantastic. Really looking forward to hearing the various presenters. So this is the VCCC Alliance Colorectal Cancer Research Showcase, and the intent is very much to showcase the extent of research across the VCCC sites, and hopefully to stimulate some discussion and collaboration flowing on from that. Certainly up front, I'd like to acknowledge the traditional custodians of the land in which myself and today's presenters and audience are located. And I would like to pay my respects to elders both past and present, and extend that respect to other Indigenous Australians present online. A couple of housekeeping notes before we kick off. So today's event's being recorded and will be available on the VCCC Alliance Open Learning page, where you can also view previous webinars. Certainly encouraging questions from the audience. So if you do have questions for today's presenters, you can do this via the Zoom Q&A function. So each presenter will be presenting for around 10 minutes, and there'll be questions directly following each presentation. And certainly encourage you to submit your questions and be part of the discussion through tonight's proceedings. So the first session will be chaired by Professor Neil Tebbert. Neil's very well known to everyone's Director of Medical Oncology at ONJCC, a long history of successful trial-based and other research. So he'll be chairing the first session, and then Associate Professor Jeannie T will be chairing the second session. Jeannie is a medical oncologist at Peter MacCallum and a Senior Research Fellow at WeHi. So I'll start the session for tonight and share my screen. So I'm presenting really on behalf of many others at WeHi, so this is certainly a collaborative effort and focusing on a broad range of both clinical and translational research. So firstly, as we all know, the level of evidence that we're always trying to achieve to change practice is a randomised controlled trial. That's at the top of the pyramid. Unfortunately, we're increasingly struggling to conduct clinical trials. This is recent data from the US, just over 12 million patients. And what they found in this audit was that only around 0.1%, so much under 1% of patients were treated on a clinical trial. So while there's many questions to be asked in clinical trials and importance of bringing that forward, we're not making a lot of progress at the moment. And over time, the challenge is actually increasing. There's more targets, more treatments, more cancer types as we define distinct subsets with distinct behaviour, and also tighter eligibility criteria, making it more challenging to enrol patients. So a lot of what I'm talking about tonight is registry-based research and how that could be helpful in advancing our knowledge base and improving our treatment of patients. So as background, there's many what I would call no-impact registries and low-impact registries. So registries have been around for decades, and I think really have under-delivered on the whole in terms of the impact on research. And that's for a few reasons. Often the data's poor quality, often rubbish in, rubbish out. So if you're not collecting complete or accurate data, it's very hard to get meaningful output. A lot of the time, the data is collected without any clear plan as to how to use it, and afterwards, some vague thoughts about quality research or audit or other aspects of potentially using the data, but often the registry data output is not as it should be. So at WeHi, over the last 20 years, we've been focusing on developing high quality registries, including critical patient, tumour, treatment and outcome data following the patient from diagnosis through their clinical journey. We've got a continued focus on quality, including data rules, use the data a lot, and update regularly the data and the data set. And a key part of this is data linkage to external and hospital administrative data. And just shown here on the slide is one of the data fields from our track registry, highlighting we're routinely collecting ECOG performance status, which is a critical prognostic factor, family history often not well documented, and comorbidities often also not very well documented. So it's important to collect high quality data, and that means collecting data from multiple sites. Often patients travel across different hospitals. Often some of the data is collected in the outpatient setting and also may be seen in the private rooms. And this, combined with looking at other data sets, hopefully with a well-trained and well-supervised data officer and a high-quality database leads to high-quality cancer registry data. So briefly, the history, this started back in 2000, collecting some colorectal cancer data, and certainly to acknowledge the contribution from Ian Jones and Ian Farragher from the surgical side have been incredibly supportive of this effort right from the beginning. Early on, we were focusing on using the data for a broad range of research. So projects, for example, from early stage colorectal cancer, looking at the impact of the National Bowel Cancer Screening Program on staged presentation, and then more recently looking at survival data. And then over time, looking more at metastatic disease, various approaches and outcomes, and starting to combine the clinical data with some of the biomarker data that we were looking at. So showing that RAS mutations are predicted for lung metastasis. Then as time progressed, we recruited Oliver to join the team initially at Ludwig, and that collaboration with Oliver has been fantastic and produced a lot of meaningful and ongoing outcome. So here we were starting to combine data and the tissue, mostly archival tissue, into a range of publications. From there, also worked with industry, particularly with Roche Diagnostics, Ventana and others. And again, looking at this combination of registry data with archival tissue to look at some of the promising biomarkers that industry were interested in, and also use the registry data to look at a validation series for a new immunohistochemistry antibody. Then moving on, getting more interesting and more impactful, I think, starting to look at data and tissue collection prospectively. So data collection prospectively and tissue samples collected prospectively, focusing initially on blood collection. And Jeannie's now led a series of studies looking at circulating tumour DNA in specific patient cohorts, which has led to some good publications. Like many of the biomarkers that we have, ctDNA is a very promising biomarker, and discovery happens often quite quickly, but the key really is to move on to validation and demonstrating the clinical utility and the clinical impact. So Jeannie has now led a series of clinical trials where we're actually looking at collecting ctDNA data and using that to inform patient practice. And the first of these, the stage 2 study, we'll be reporting next year, which is an exciting time for all of us. Also other tissue-based research, so working with Oliver and Margaret as the leads of the recently recruited forecast study. So that's an organoid-based study looking at using organoid sensitivity to guide chemotherapy decision-making. And others in the group, Shahara leading a study looking at EREG and AREG in colorectal cancer, and Vanessa looking at trying to define a patient cohort that may benefit from an exciting and promising new drug that's becoming available. Again, a bit of the hare and the tortoise, so well-established path for new drug development in oncology, very good at going through that phase 1, phase 2, phase 3 development, defining new drugs. But a lot of questions remain often after new drugs are developed, particularly around the optimal patient to treat, optimal duration of treatment, optimal sequencing, optimal combinations. And many of these questions are important, but until recently, we really haven't had a good mechanism for pursuing these in a research setting. So we've developed the concept of prospective interventional studies, what we're calling registry-based randomized controlled trials. So the concept here is the registry collects the relevant patient treatment and outcome data, and patients are then entered and randomized into various clinical trials. For example, the OLD-TRACK study that Vanessa's leading, looking at alternating doublets of chemotherapy for metastatic colorectal cancer, which is recruiting well, along with some other registry studies, including the Resolute study that Jeannie's leading that will soon be opening. Starting to move into new directions, and as the group grows and the interest grows, we're able to use the data further to leverage into other directions. For example, Richard is now doing a PhD focused on health economics and using the data to look at the impact of, for example, circulating tumour DNA on cost. Grace is doing some nice work looking at some patient education. So the concept here, using the registry data to generate patient education information sheets and improve their understanding of their treatment and the planned outcomes. Looking at collecting more PROMS data, so we've got clinical data tissue data, and now starting to collect some PROMS data, so patient data entry that will be combined with the registry data efforts. Kate is starting to look at measuring quality using the clinical registries to measure quality, and hopefully by defining some metrics, we can feed data back to clinicians and over time have a virtuous cycle where we can measure and improve quality. And then a new direction also will be clinician decision support using the clinical registry to understand treatment and outcomes in routine care, and hopefully using that to inform practice and to improve practice. So to finish up with, just to acknowledge this is very much a team effort. So I've mentioned many of the clinicians and scientists that are involved in this process, but there's also many others that are part of this: research nurses, project officers, lab managers, etc. Some are shown on the slide, some aren't, so apologies for anybody that's not on the slide, probably because I couldn't find your photo. So just to summarise the effort at WeHi, over time we've built up a large collaborative research effort. It's all based around high quality registry data. So increasingly that's expanding, not just in Melbourne, but sites across Australia and internationally, and wherever possible looking to use that data across many different research projects, combining it with tissue-based analysis and particularly focusing on developing new research directions. So I'll finish there and happy to take questions if there is any.
So thanks, Peter. So just to remind the audience to ask questions, we've got a question now. And the question relates to quality. We'll maybe expand on this a little bit. So I think this relates to quality indicators, right? It's not quality of life that you're assessing there. So tell me, what are the sort of key quality indicators that you're collecting, and have you always been collecting these indicators, or these things that something that's evolved as time has gone on?
Yeah, so the intent's to use the data that's already been collected and look at indicators at diagnosis. So do patients have appropriate biomarker testing, RAS testing, BRAF testing, MR testing, which is informing practice, looking at the appropriate treatment. So if you're RAS wild type left-sided, do patients get EGFR inhibitors through the course of their treatment? I think one of the interesting aspects there is that we do see a large variation in outcomes across hospitals. The median survival varies by more than about 15 months, looking at the sort of best performing versus worst performing hospital. So it does suggest that there is variation in quality, and looking at also surgical intervention. So for example, one of the interesting findings, there's quite a marked variation in liver resection rates for colorectal liver metastases, and that actually looks like it correlates with survival. So it's trying to define some metrics, very challenging area. I think we're just at the beginning, but hopefully we can have an impact there.
Okay, so and then I might ask another question, and obviously you've sort of got a lot of data out of the registries over time. But it still requires a certain amount of resourcing and effort, and I think you've leveraged pharma support for some time to do that. How do you see the future with the sort of increasing digital innovation, use of electronic health records? Do you think that this will continue to be the solution, or will be using sort of methods of extracting data from the various electronic records that are increasingly in use, whether that's pathology or clinical data sets?
Yeah, it's a very important question. I think, as you've hinted, the cost of collecting data and data managers and maintaining that whole ecosystem is substantial. I don't think it's sustainable, particularly looking at multiple hospitals. So ultimately, it would be great if we could extract data that's already there. The key is obviously that the data is collected in a standardized fashion and it can be reliably extracted. Things like ECOG performance status comorbidity is often not well documented. So some data I think we'll have to keep trawling for, but many of the data items over time, hopefully we can extract directly from the medical records. So I think ultimately it'll be a hybrid model, and over time more and more data will be extracted and the data sets will be auto-populated, but then supplemented with additional data that's extracted from other places.
Right. Okay, so I don't think there are any more questions, so we might move on to the next set of presenters. Thanks very much, Peter, for a great summary of work over a number of years. So the next two speakers, so we've got my colleagues, John Mariadason and Fiona Chionh, who work with me at the ONJ Cancer Centre, and they're going to talk about, so that's a medical oncologist and scientist, and they're going to talk about some of the colorectal cancer research underway at the ONJ Centre. I don't know who's going first, but out of the two of you, but John and Fiona.
That'll be me, Neil. Okay. Hopefully you can all see my screen. Thanks, Peter, for putting this initiative together. It's a great opportunity for us to showcase what we're doing actively in colorectal cancer research here at the Austin. So this is us here, located, it's a major teaching hospital based in the northeast of Melbourne, not too far away from the Parkville precinct. What I'd like to do today is briefly showcase, I think, what the active colorectal cancer research we have underway. And what I thought I'd do is, briefly introduce the various departments that are researching this disease, introduce some of the key researchers involved and some of the techniques and approaches we're taking that hopefully will be of use to the broader community here in Melbourne. So just to kick it off, the first department that came to mind that I wanted to emphasize was anatomical pathology, and we have a number of very active academic pathologists in the department. And David Williams is probably the one leading the colorectal program. He's had a long interest in this disease, having done his PhD at the Ludwig. But what David has done is really assembled very nice and large data colorectal cancer samples and assembled these nicely into a TMA and invested quite a bit of time in importantly getting the survival and outcome data associated with that. This has been a very valuable resource that is shared with a number of investigators and enabled a lot of prognostic markers to be identified. More recently, as David's been moving more into the use of AI for obtaining even more, data from pathological slides. Medical oncology, Peter introduced Neil, who heads the unit here at the Austin. It covers a range of cancers, but has a very active GI, sees a number of GI cancer patients from the Northeast catchment. As mentioned, Neil has run a series of investigator-initiated trials ranging from early phase to larger phase 3 studies. And we'll touch on, Fiona will touch on how we've actually done a series of biomarker studies with those trial cohorts. Radiation oncology is also is sort of an increasing area of research activity since the installation of the MR-Linac here at the Austin, and that's allowed us to recruit Stephen Chin into the department, and Steve's running a series of trials now looking at using the Linac for dose escalation studies, as well as for identifying predictive markers of rectal cancer response to radiation. Very important to mention the Department of Surgery, which is the University of Melbourne. So the University of Melbourne has a large presence here at the Austin through the Department of Surgery and Medicine. The Department of Surgery has had a very long history in colorectal cancer research, really led by Prof. Christofi for many years, but now by Prof. Murali and Marcos. Alex Dobrovic has also joined the department recently. And they've spent a lot of time working up elegant mouse models of colorectal cancer metastases and use these models to test new therapies, as well as now more recently looking at biomarkers of colorectal cancer metastasis likelihood, And finally, it's the Olivia Newton-John Cancer Research Institute, which are also based here at the Austin. We have about 150 researchers, a very active GI cancer program, including colorectal. That includes T. Ernst, who's our director, and his research interests are in targeting not just tumor cells, but the tumor microenvironment. There's my own group. We have a focus on epigenetics. And recently we've recruited Lisa Milkey into the Institute who's interested in different immune subsets and their role in progression of the disease. Andrew Scott's been here for many years, and he's been developing a series of antibodies, both to image colorectal tumors as well as target them. And he's particularly interested in the EGFR pathway. I'll just finish by mentioning some of the resources that we have. We have a large panel of cell lines that we've genomically profiled, which are very useful for drug screening. And we're very happy to share those and our expertise with those models. And the other really, I think the resource that we have is genetic mouse models. So we have a series of mouse lines that I think cover all of the major pathways that drive this disease: Wnt signaling, MAP kinase, microsatellite instability. So they're very useful tools for crossing to additional animals if you want to work out the role of a particular gene in this disease. So hopefully that gives you a little snapshot of what we do and some of the collaborative opportunities that we have. So I'll quickly stop and hand over to Fiona, who'll continue.
Thank you for the opportunity to talk. This evening, I'll provide a brief overview of some of the work from the Translational Gastrointestinal Research Program at the Olivia Newton-John Cancer Research Institute, which is co-located with the Austin Hospital. Led by Professors Tebbert and John Mariadason, who you've just heard from, this program has attracted 6 medical oncologist doctorate students to date and has a very strong focus on colorectal cancer as well as upper gastrointestinal cancers. The first major translational research focus of the program has been to identify biomarkers of response and resistance to targeted therapies for colorectal cancer. This approach utilises patient clinical trial specimens and outcome data from prospective clinical trials to perform biomarker sub-studies. And the ideal study design here is a randomised trial so that patients who receive the targeted therapy are comparable to those who do not, and have a control arm without the targeted therapy so that the baseline prognostic impact of the biomarker can be taken into account. Identification of predictive biomarkers may enable patients who are unlikely to respond to commence alternative treatments earlier and also avoid unnecessary side effects and costs. And at multiple steps along the way, collaborations with trials groups, other clinicians, scientists, and other researchers are paramount. The MAX clinical trial, led by Professor Tebbert, adopted this ideal study design for retrospective biomarker analysis. This was an investigator-initiated multi-centre phase 3 randomised controlled trial of first-line treatment in metastatic colorectal cancer. And patients were randomised to receive treatment with capecitabine chemotherapy alone or in combination with the anti-angiogenic agent bevacizumab plus or minus mitomycin C. The MAX study was positive for its primary endpoint of progression-free survival, demonstrating a PFS benefit for the addition of bevacizumab, and 83% of participants consented to biomarker analysis sub-studies. Not all patients respond to bevacizumab, with modest survival benefits reported in unselected populations. Therefore, there is an urgent need to identify predictive biomarkers to bevacizumab response. And our laboratory has now completed several biomarker studies from the MAX trial to address this, and have found that low VEGF-D, IHC expression is predictive of bevacizumab PFS benefit, that VEGF family single nucleotide polymorphisms are novel biomarkers of prognosis and hypertension. And the third biomarker study, led by Dr. Jennifer Mui, investigated whether the transcriptome-based consensus molecular subtypes, or CMS classification, was predictive of bevacizumab response. CMS was found to be prognostic for overall survival, so patients who were CMS2 had the best overall survival, and patients with CMS1 tumours had the worst overall survival. Importantly, CMS was also predictive of PFS benefit from bevacizumab in the CMS2 group and possibly the CMS3 group, which had smaller patient numbers. The second major translational research focus of the program has been to address the clinical need to overcome resistance to existing therapies for colorectal cancers. And this approach utilises data from preclinical studies identifying mechanisms of inherent and acquired resistance to existing therapies. This data is then used to inform the development of new treatments and treatment combinations with the goal of testing them in clinical trials. And biomarker translational sub-studies incorporated into these trials can potentially refine clinical utility and provide us with a deeper understanding about molecular mechanisms of response and resistance. And we have used this approach in the VEDA study, which is due to commence shortly. In the clinic, the addition of EGFR monoclonal antibodies to chemotherapy or best supportive care improves survival outcomes and response rates in patients with RAS wild-type metastatic colorectal cancers. However, one of the barriers to efficacy is that EGFR monoclonal antibody treatment does not consistently induce apoptosis in KRAS wild-type colorectal cancer cells. We found that when you treat these cancers with EGFR inhibitors, they do induce the expression of pro-apoptotic BH3-only proteins. However, despite this, apoptosis is not induced and only a cytostatic effect is observed. This suggests that EGFR inhibitors may prime these cancer cells for apoptosis. However, they may be insufficient levels of pro-apoptotic proteins to overcome the threshold for apoptosis. We also found that histone deacetylase inhibitors induce robust apoptosis in colorectal cancer cells, and that the pro-apoptotic activity of these drugs is driven by the increased expression of pro-apoptotic BH3-only proteins and decreased expression of pro-survival proteins. We next found that regardless of the MAP kinase genotype of the colorectal cancer cells that are treated, when you combine a HDAC inhibitor with either tailored MAP kinase inhibitor treatment or MEK inhibitor treatment, we observe an increased expression in the pro-apoptotic proteins and synergistic induction of apoptosis. Specifically, when we treat KRAS wild-type colorectal cancers with a combination of a HDAC inhibitor, sodium valproate, and an EGFR inhibitor, we observe significant induction of BIM and BMF, induction of apoptosis, and significant inhibition of growth of KRAS wild-type colorectal cancer xenografts in vivo. And based on this preclinical data, we have developed the Beta clinical trial, which is a randomised phase 2 trial of first-line maintenance treatment in patients with RAS wild-type metastatic colorectal cancer. So in this study, patients will be randomised to maintenance EGFR monoclonal antibody alone or in combination with the HDAC inhibitor sodium valproate after completing 4 months of treatment with chemotherapy plus EGFR monoclonal antibody. And the primary endpoint of the study is progression-free survival. We have incorporated translational biomarker studies into this trial and will collect both tissue and peripheral blood samples to answer research questions, including are changes in histone acetylation in PBMCs predictive of improved efficacy, and are potential resistance conferring mutations in ctDNA associated with efficacy outcomes in patients treated with EGFR monoclonal antibody maintenance. We are excited about the opportunity to collaborate with other colorectal cancer clinicians to test this combination in the clinic and about the scope for further collaborations in the incorporated translational biomarker studies. So thank you for listening, and these are my acknowledgements and funding.
Okay, well, thanks very much, Fiona and John. I'm getting my video started again, thank you. So it's open for questions. You can put your questions in the chat, but while we hopefully wait for a question or two, maybe we can, I can ask, this can be either or both of you. So the CMS, the consensus molecular signatures was, people had a lot of hope that that would be useful. How do you see that playing out into actual clinical practice? Do you think that this will be something we'll be evaluating in patients to decide on treatments? And do you think it's something that changes during the course of the illness or its treatment?
Yeah, so I'm happy to answer that question. This was the study led by Jennifer. But I can sum that up by saying, look, I think it's very promising data, but I think there still needs to be quite a bit of work before this reaches real time in the clinic. And that's for several reasons. So firstly, the data does need to be validated in an appropriate clinical cohort. There have been CMS biomarker studies performed in the FIRE3 study and the CALGB 80405 study, and the issue there is that these were different clinical populations and different treatments compared to our study. So we didn't have a KRAS wild-type selected population, and the other studies had combined chemotherapy and examined cetuximab versus bevacizumab. What I can say about that is that the prognostic data from those studies is concordant with the data that we have found, But in their predictive analysis, really the question has been, look, has it predicted the benefit for bevacizumab versus tuximab? Whereas our question is, does this predict the benefit of bevacizumab? So I think this still needs to be validated in a second similar trial cohort. So the second issue is with regards to the actual classification of the CMS subtypes. So the CMS, what we call the predicted CMS classification, leaves up to 13% of the tumours unclassified, an unclassified group. And this may be due to having a mixed subtype or because they just don't reach the cutoff for subtype classification. And how Jennifer and the team dealt with this is the primary analysis was performed using something called the nearest CMS classifier, which places the tumours into the nearest CMS subtype to try and eradicate that unclassified group. They did perform a sensitivity analysis using the predicted CMS classifier, which showed similar results, but the issue remains that the CMS classifier does need to be refined further, or another option is that further clinical cohorts need to perform sensitivity analyses comparing the predicted CMS classifier versus the near CMS classifier. And then, as you touched on before, the third point is that we need to do more research looking at how the CMS subtype evolves when you go from the primary tumour to the metastatic tumour, and even metastatic tumours that are treated in different ways. And this will be needed to give us confidence to apply the CMS subtype to tumour samples taken from those different stages.
Okay, thank you. So we've got a couple of audience questions now, which we can cover live, and then I think that we'll move on to the next talk. So these are both for John. So the first question, which is kind of a follow-on, have you been able to characterise the cell lines according to CMS subtype? And then a second question, with the cell lines, have you compared behaviour of cells grown in 2D versus 3D and how they behave in response to treatment?
Yep, sure. I'll try and be quick. Yes, we have done the CMS classification. Actually, Oliver Sieber did that, and a lot of the cell line characterization has been in collaboration with him. And we can identify the different subtypes, except for CMS4. And that, I think, makes sense because that seems to be really a stromal-driven signature when you use patient samples, and that clearly not going to be the case when you use cell lines in vitro. We have done a lot of studies comparing 2D versus 3D as well as xenograft growth. So we've grown all our cell lines as xenografts. Interestingly, a lot of the features are retained. So certainly histological grade that we see in vitro can be recaptured in vivo. But yeah, I completely acknowledge that there are different growth behaviors when you do move these cells around in terms of different growth conditions.
What about in response to therapies? Do you see any difference there, 2D versus 3D?
Look, I think we haven't done a lot in that. I know Oliver has done some, and now he's doing it. I think cell lines are now being superseded by organoids to a large extent. I personally don't think so. I think after working with cell lines for all of these years, I think they're still very good models, and I don't see grossly different responses or behaviors. I mean, there are subtle ones, but I don't think things change dramatically when you change 2D versus 3D. I guess, but it's a magnitude issue. Okay,
great. All right, well, thanks very much, John and Fiona. So we'll move on to the next speaker. So it's a pleasure to introduce Ian Hayes, who's head of the colorectal surgical unit at Royal Melbourne Hospital, and he's going to give us an update on outcomes of research projects in colorectal cancer at Royal Melbourne Hospital and potentially other centres. Thank you, Ian.
Okay, that's great. I'm just trying to share my screen. Share, here we go. Okay, thanks very much for having me. You've been hearing from big resource groups. Now you won't be hearing from a big resource group. But since 2014, we've been a small but serious research group. So our initial introduction to this was through the Dr. Foster Global Comparators Group in 2014. We were asked to go to one of their meetings, and what they allowed us to be involved with was an international group of clinicians interested in research. They allowed us access to administrative data, and they importantly allowed us access to data analysts. And we realized that from this, that we could start a research project without any resources. And on the basis of this, we dialed up the colorectal research fellow at the Royal Melbourne to link them to a higher degree. I involved Anita Skandarajis, my co-conspirator, to give us a bit more research cred. And our program has been very successful in terms of producing degrees. So we've successfully completed 2 doctorates, 2 masters, and we're on a third doctorate at the moment. Our focus has been administrative data, and I should talk a bit about what this is. So administrative data isn't the same as clinical data. Every patient who goes into hospital has the events of their admission categorized into alphanumeric codes. That's done in a very systematic fashion. It's highly regulated, it's reproducible, but it ain't exactly the same as clinical data. So what we call an apple, they may call an orange, but they'll call it reproducibly an orange. And furthermore, they may not actually have a word for apple. You can't use admin data out of the box. You've got to gain an understanding of it and work out how it correlates with clinical data. It only relates to events that happen during hospital admission. It doesn't give you any outpatient data, and it doesn't give you any survival data. The other angle that we've pushed throughout all this has been improving our research methodology. When we were initially involved with the data analysts through Dr. Foster, which has since folded, I didn't know what they were talking about. I didn't know what I was talking about. And importantly, they didn't know what I was trying to talk about. So I went off and did a Master's of Epidemiology, and on the basis of that, that's led to another arm of research, which is my 2 supervisors, Dr. Eliza Melanzi and Dr. Jeanette Rees, have kind of stayed interested with me for some reason. And we've done some very gnarly studies through the highly granular data of BioGrid. This is sort of a broad brushstroke of the admin data we've produced through our unit. We started simply first looking at in-house Royal Melbourne admin data and correlating that with clinical notes. Then we moved to population level data, which is the Victorian admission episode data set, which is the whole data set for the state. First studies of that were led by Dr. Foster analysts, which was fairly simplistically from a methodological point of view. We've subsequently got more complicated and started to do data linkage. So linking to the death index allows us to do overall survival. Our methodologies increased with logistic regression analysis and proper survival analysis. This year, we're linking, in addition to the cancer registry, which essentially gives us very good path data. And going forward, we're planning to link to Medicare, which will actually give us some oncology drug use data. In all this data stuff, we've actually published quite a few things about the use of data. The highest level we got up to is in JAMA surgery. So just a few things about how we use the admin data. So we've concentrated on creating algorithms of admin data to more closely resemble clinical data. How we started with this for cancer was that we used BioGrid for case finding and created a list of cancers. Then we generated a list of cancers from admin coding with ICD-10 coding. We matched the 2 lists and then finally matched both lists against clinical notes and used that for our accuracy studies. Essentially, with the accuracy, we found that if we combine codes both of location of tumor and what was done with the tumor, we lose a bit of granularity but gain greater accuracy. So we work with right colon, left colon, and rectum, which are reasonably accurate. This was a quick view on an excessively detailed slide of the first project we did using state level data. And I'll take you through it, but essentially it was a fairly simplistic study, essentially a narrative study, but this ain't out there anywhere else. We found that for this 6 or 8 years of looking at it, most of the colorectal cancer in Victoria is done through the colorectal training hospitals, which are 8 or 9 hospitals within Melbourne. However, within Melbourne, the non-training hospitals, they do about 1/3 of the cancers, but they do the same spectrum of cancers as the training hospitals. And the big inner regional hospitals, such as Ballarat and Bendigo, do a smaller number, but the same spectrum of work. But interestingly, the small inner regional hospitals do a lot of the cancer. They do about 1,000 of the cancers, and they do 250 of the rectal cancers. Sorry, I've gone on one too far. This study we were very pleased with because this was the first time we achieved our goal of being able to provide population level comparative studies using decent methodology for the whole of the state. We looked at both the short-term outcomes comparing regional hospitals to city hospitals and the long-term outcomes comparing city hospitals to regional hospitals for colorectal cancer. And essentially we found that short-term outcomes were similar across city and country, and similarly the long-term overall survival outcomes were the same. Of course, there's a selection bias whereby some of the more complicated cases go to the city, but overall the comorbidities of the patients in city and country were similar, which was surprising. Next study I'd like to highlight is one where we entered into the dizzy world of propensity scoring. So here we did probably the largest pseudo-randomized study looking at LAP v open. So we took the whole of the state's data. We concentrated on right colon cancer because we knew the admin accuracy was very good, and we also felt that it would be the most widely used resection. I'll go back to that for a minute. We ended up with about 2,000 in LAP group and in the open group. And because it was propensity scored, it's pseudo-randomized. We analysed it in a number of ways, but essentially in terms of the short-term outcomes, we found that the laparoscopic surgery was less likely to give you a prolonged length of stay, less likely to get you a return to theatre, and more likely to get you discharged home. In terms of overall survival, it must have moved a bit too far, no difference. So LAP versus open, no difference in overall survival. We haven't been able to coerce anybody to publish that yet. While we're looking at long-term outcomes, we meta-analysed the long-term non-oncological outcomes for colorectal cancer, looking at adhesional obstruction and incisional hernia. There was 15 studies and there's 6 RCTs that got published in Diseases of Colon and Rectum. And basically, LAP surgery produced fewer incisional hernias and fewer obstructions. But if you looked at cases actually requiring surgery for those complications, there was no difference between LAP and open. Now I want to move to a very different area from the admin data. The admin data is all about using population level data to produce fairly clean studies. What I've done here working with people from the School of Population Health is, I've got a funny button here. We've tried to get detailed and complicated using the BioGrid data. BioGrid's great data, but trying to do complex studies from any database poses methodological problems. First thing we wanted to look at was the use of neoadjuvant chemoradiotherapy for early stage cancers of the lower part of the rectum. The literature is unclear about whether this should be used. Certainly it should be used for T3s, but we're not sure for the earlier T stages. And in the lower rectum, there's much less mesorectum. So we worry that an earlier stage tumor may be closer to the resection margin. So what we did is we took all the BioGrid low rectal cancers, which were less than or equal to T2 in histology, having a TME. We found 189 of them did have long course chemoradiotherapy and 136 did not, and we compared them. But I wanted to highlight some of the methodological problems. So the MRI data set was incomplete. We only had it for about 2/3 of the cases. MRI is inaccurate for these cases in the first place. And unfortunately, it's a good prognosis tumor, so you've got very few events to compare. So there's only actually 5 local recurrences, 40 recurrences of any sort. And the analysis got into the complex world of time-varying multivariable analysis. Bear with me while this gets more and more complicated. So when we did the time-varying analysis, we found for the first year, people who had chemoradiotherapy actually had higher recurrences. It eased off, or they equalized in the subsequent 4 years. And that raised some weird questions. So we would have thought the people having chemoradiotherapy would have fewer recurrences. We tried to solve this by doing a subgroup analysis where we looked at people at a smaller cohort where we actually had MRI data showing that they were less than equal to T2 to start with and less than equal to T2 on histology. So thus people who probably didn't have any downstaging. And in those cases, there was no difference in the survival. So the increased hazard of recurrence only applied when we hadn't adjusted for downstaging. And that brought us to the next study, where we started looking at the concept of complete pathological response. So the clinical context here is that people who have complete pathological response seem to have better survival, but we wondered if people who have a partial response have a sort of dose-dependent, not dose-dependent, but a trend towards better outcomes. This study had a whole lot of complicated data issues. So the first thing is that the initial staging was with MRI. MRI isn't completely accurate. Your subsequent staging is with histopathology, which is comparing kind of apples and oranges. Mostly in Victoria, people don't have post-chemotherapy MRI scans to compare the initial one to. And tumor regression grade as an index of downstaging isn't great, it's pretty subjective. And for lymph nodes, it's a disaster. So our solutions were firstly to try and sort out the accuracy of MRI. So what we did is we looked at people who didn't have chemoradiotherapy and did an accuracy study comparing MRI to their path, which wasn't interfered with by any downstaging. That gave us an accuracy for T3 of about 80%. Then you've got the question of how do you put that 80% into your main cohort? And we solved that by bootstrapping it, which meant that we ran the 80% 10,000 times through our case series to give us an estimate and confidence intervals, and then started the study. We chose to look at T downstaging rather than tumor regression grade on the grounds that that was much more objective than tumor regression grade. And nodal status is hard to assess on MRI, and it's hard to assess partial nodal downstaging. So we simply adjusted for nodal status. So the study was people starting of an MRI that said they were T3, less than 12 centimeters from the anal verge. They all had neoadjuvant chemoradiotherapy, and they all had a TME. And they were all fairly recent, so that it was good quality MRI. We had 528 patients. We found that about half of them had no downstaging on T-stage, about a third had partial downstaging, and about 20% had complete downstaging. And to our surprise, we found no effect. So partial downstaging showed no evidence of a true difference. And similarly, complete downstaging of T-stage showed no evidence of the difference. And that was adjusted for just about everything we could find in BioGrid. The next study looked at another topical area, which is the association between nodal count and long-term oncological outcomes for right-sided cancer. Certainly, extended lymphadenectomy is very topical at the moment, but perhaps we're jumping ahead of ourselves because the evidence base for the association between lymph node harvest and recurrence isn't fabulous. So again, we did a BioGrid study. We took all stage 1 to 3 adenocarcinomas. We confined it to the cecum and ascending colon and confined it to patients having a standard right hemicolectomy to decrease the amount of heterogeneity in the study, and that gave us 1,300 patients. We categorized the nodal harvest with a control level of 12 or less, And then we went up in ascending categories of nodal level of 13 to 15, 16 to 20, and greater than 20. And we stratified by nodal stage because there's some evidence that the results are different at different nodal stages. And we did a Cox analysis of this. And to cut a long story short, these are early data, but weirdly, we found no difference. So we're certainly finding no evidence that there's a trend that as nodal count increases, that survival and recurrence change. And that's been stratified for all levels of nodal status. That's not published yet, but it's interesting in the face of increasing interest in extended lymphadenectomy. I'll close up with a slightly ironic line. The stuff we do sometimes seems very tantalizing. A lot of the time it falls on its face. We've worked with a number of different databases. None of them is perfect. No study is perfect. Databases are just tools, and they're really a framework of potentially confounding variables that you need to find ways of using. It's really important to understand the strengths and weaknesses of the database and to be aware of the analysis options to allow your best study design. And I suspect going forward, the best cancer questions and maybe the answers will come not just by comparing things within the data sets, but looking things from outside our cancer data sets and basically using the data sets as a set of confounding variables to allow you to do a better analysis. Thank you very much. Thanks very much,
Ian. Obviously a very significant body of work over quite a number of years. We're unfortunately run out of time for questions, so I'm going to move on to the next speaker, who's Rob Ramsay. So he's going to close this session talking about real-world gaps in managing colorectal cancer. Thanks, Rob.
Thank you, Neil, and also thank you very much, Peter, for allowing us to all get together, because normally we don't see very much of each other except on Zoom screens. It's also very helpful to hear Ian, who basically set the stage for some of the things I want to talk about. One of the key points that I want to make is that the way you conduct your laboratory research is sometimes dictated by the philosophy that you bring to it. And one that I've built upon with my colleagues is that every part of the patient journey has an opportunity to perform research and understand what's going on with our patients. And I think ultimately all of these areas can be improved upon. And if you think of the circle of the patient journey, at all points, I think we can do better. And the way we do better is to do things like Ian's been talking about, where we actually understand what's happening to our patients and what we're doing to them, and identifying opportunities to engage people like John and myself, who are laboratory researchers. At Peter Mac, we do only some of the things that are part of GI cancer, and that's probably true of all the members of the Alliance, the VCCC. It's important to understand who comes in the door, so that can guide what you might focus your attention upon. We tend to spend a lot of our attention on patients with advanced GI cancer. And indeed, we do more rectal cancer than colon, even though in a research sense, we often combine colorectal cancer together as a single entity. But indeed, that's not the way they're managed, as highlighted by what Ian was talking about before. The other thing we do that's of potential distinction is we manage patients with peritoneal cancer, that is spread of cancers to the peritoneum, and these metastases are managed quite differently to probably any other kind of GI cancer, involving surgical cytoreduction and a rather unusual form of chemotherapy. Also, this centre tends to have referral base for patients with squamous cell carcinoma of the anus, which is basically at the end of the GI tract. and this requires quite complicated surgery in some cases too. And finally, a substantial group of our patients, as with many in our alliance, come to us with liver metastases. So that governs the kind of research you might think about doing. So let's just focus on the peritoneal cancers for a moment. We became very interested in this. This is a kind of cancer which occurs on the abdomen. You can probably see the nodules on the micrograph on the side. There's many of them. They're hard to manage, and they're removed surgically and then treated with chemotherapy up to a point. And if there's too many, then other options are considered. So we were very interested in what happened to those particular cancer cells when we removed them from the patient and asked whether they responded to the drugs we normally treat those patients with. And on occasions, you have a patient, as the one shown here, where all the drugs that we normally use basically don't work on this particular cancer group. And that led us to do a medium throughput screen, looking for other drugs that might work better in such a patient. And this is an example where a drug which is used in another indication allowed us to kill the tumor cells in the test tube. And that allowed our medical oncology colleagues to consider using this drug in this particular patient. And although it wasn't a huge success, it certainly produced a response. And it was a beautiful proof of principle that information that we generate in the laboratory can sometimes inform our clinical colleagues to change the treatment of those patients. This is an important problem for us because peritoneal metastases is very difficult to manage. And indeed, it's even more complicated than that. You might have noticed how many different lesions there were on that micrograph before. And if we take individual lesions, grow them up, and ask which ones are sensitive to which drugs, it becomes quite evident there's a lot of heterogeneity in those particular tumors. So some respond and some don't. So we've got to do a lot better in this space. And this is something that we're working very heavily along the lines of using organoids to inform our research. And Mike Flood at the moment is pushing along this area of research. Another area that's of great interest to us is the way the immune system interacts with cancer cells. And indeed, we've been able to grow rectal cancer organoids, mix them with lymphocytes from the same patient. And we've been able to identify at the beginning of the treatment journey, those patients that are likely to do best with a standard of care. And those that have got the best immune function do best in our clinic. So the immune system is a very important arbiter of outcome of our patients. Similarly, we've been able to embark upon growing organoids from patients with anal squamous cell carcinoma. We've probably got the largest series of anywhere in the world. And the work of one of our PhD students who's now a fully card-carrying consultant colorectal surgeon, Glenn Gira, he did this beautiful study generating a range of lines, allowing us to explore a whole lot of biology of patients' squamous cell carcinoma. We've extended this now into other members of this category of cancer in the peritoneum, and these include penile cancers, vulvar cancer, and vaginal cancers. And indeed, we've been able to think of new therapies which might help these patients. To think about this whole journey, we need to not only think about drugs, the chemotherapy, the surgery, but also the way we manage anesthesia. And one of my colleagues, Bernard Riddell, who's head of anesthetics here, is very interested in the impact of the choice of the anesthesia that we manage our patients with when surgery is conducted. And it seems quite clear from early data that that style of anesthesia is very important and affects patient outcomes. And Bernard's got a very large clinical trial in progress to explore this question in greater detail. We're very interested in surgery, and indeed most of my lab has some surgery connection. Quite a lot of the fellows in the lab are surgery trainees or indeed already card-carrying colorectal surgeons. And we've been interested in the simple process of blowing up the abdomen for laparoscopic surgery, asking the question, about the gas that we use. And the gas that's typically used is carbon dioxide, comes straight out of the bottle. It's very, it's clean, but it's dry and desiccating. And a whole lot of animal studies have suggested to us and others that if we warm and humidify that gas, we can protect the peritoneum from the consequences of that desiccation. Furthermore, when there is that damage, cancer cells like to grow on that damage. That initiated a parallel effort to conduct a randomized clinical trial, asking the question, what's the difference in laparoscopic surgery cases with cold dry CO2 versus humidified warm? We've finally recruited enough patients in this study, and this is driven by a colleague, Craig Lynch, who works at the Epworth. And this study has given us some very interesting clues. The humidified gas leads to a reduced inflammatory response in patients in the laparoscopic group, and a smaller group of patients still provided enough information to tell us that the induction of this C-reactive protein, an inflammatory marker, is similarly reduced if you bathe the abdomen with humidified warm during conventional open surgery. And this work has been driven by a fabulous RA in my lab and one of our colleagues who retrained and is now a consultant colorectal surgeon, Tim Chittleborough. If you look at the abdomen, you can measure damage by using a technique called scanning electron microscopy. It allows us to look at the fine details of the cells that line the peritoneum. And indeed, what we found, like we found in the mouse and in the pig, that the humidified warm gas reduces the amount of damage seen inside the abdomen, and this damage increases over time. Making the point that rectal and colon cancer patients have to be managed differently, when we introduce the humidified gas into those two different groups of patients, we're able to find an improved response in those patients with the humidified gas, such that their length of stay was significantly reduced. And this is good for everybody, the patients, the health system, and of course, we want our patients to go home well very quickly. You'll notice there's a unifying theme here that we're very focused on ways of reducing inflammation in our patients. So the take-home messages that I want to get across here, which are true for all of our partners within the Alliance, and that is, we need to identify where the needs are to improve the patients that we care for. We need to work with a team that actually provides that care, and I'm very blessed to have that opportunity. We need to be able to enthuse and train the next generation of researchers, and that includes not only our lab-based, but our clinical colleagues. For me to be able to do this talk, I'm supported by a fabulous group of colleagues, a large clinical team, and a great laboratory team. I've highlighted some people in red just to bring them out a little bit. They're not more important, but they're very precious to the opportunities that we've been able to take advantage of to explore what's going on in our patients. So I hope I've caught up a bit of time. I hope I haven't spoken too fast.
So thanks very much, Rob. So bang on time. I don't think there's any questions in the chat. Otherwise, we're going to do one question and finish the session exactly on time. So as expected, you're covering everything: anaesthesia, surgery, not much basic science in there, but it's clear where your interests are. So anyway, if you could only do one more thing, one more study, what's it going to be?
I think the biggest contribution my team could make would be to improve the management of patients with peritoneal cancer. I would also make a little point, Neil, that I gave the helicopter view of it, but underpinning all of those organoid studies is genomics and immunoprofiling and the like. And I think for our audience to get an understanding of the big picture questions, that's the way I put it together. But I'd like to leave, I suppose, this business of running a research team, improving the outcome of our patients with peritoneal cancer. That excites me a lot. I think we can do something very useful in anal cancer. We've got a study that's, I think, is going to run real time next year. And obviously the rectal cancer patients need to get the best out of the laboratory research as well. So I really don't have a favorite child in this. Neil, I love all of the things we're doing, but I think I'd focus on the one I mentioned most.
Okay, great. Well, we're bang on time, so we've got a 5-minute break. Dogs go crazy in my house, and we'll meet again 7:40. 5-minute break, and we'll start the second session. Thanks very much for your attention.
Hi, Jeannie.
Hi.
Glad we had a break because there's quite a change of focus. My stuff, but that's good. Okay.
It's good. It's good to have a different focus.
Yes, yeah, it's good. Can't remember who else is talking in this session, in this segment.
Yep. So I will introduce. I'll get started.
Oh, yeah. Okay.
Yep. Hi. Thank you, everyone, for coming back to this session of the evening. So I'm Jeannie T. I'm a colorectal oncologist at the MacCallum Cancer Centre and a research fellow at the Walter Eliza Hall Institute. So we've heard an impressive array of multidisciplinary and importantly collaborative research in the previous session. I'm going to change tack a little bit in the second session with a slightly different focus of research. We're going to hear from 3 speakers about psycho-oncology research, about big data research, and finally on health economic modeling in biomarker-driven precision oncology era, before I hand back to Peter to wrap up. So the first speaker for this session is Associate Professor Suanne McLachlan. Suanne is a medical oncologist and Director of Oncology and Cancer Services at St Vincent's Hospital, Melbourne. She has a long-standing interest in reported outcome research and will speak to us about psycho-oncology research for patients diagnosed with early-stage colorectal cancer in the era of personalized medicine. Suanne.
Thank you. Can you see that okay? Yeah, okay. Thank you for the invitation, and I'm glad we had a little break between the sessions because this is quite a change of focus. So at St Vincent's, I guess we have a bit of an interest in what I term patient experience research. So these are patient-reported outcomes, psycho-oncology type studies, and we often get the questions from the bedside, and they come up in our practice, and we look at them and bring them back to our research dry lab, if you like, and work them up in research studies. So this concept of the fear of cancer recurrence actually came to me when we started participating in the dynamic studies through at St Vincent's, Jeannie's studies in colorectal cancer. And you know, talking to the patients, that really struck me that what would it be like to live with this knowledge of having circulating tumour DNA in your blood, and how would you cope with that going forward? So I had a bit of a light bulb moment and spoke to Jeannie, and so I'm very grateful that she was willing to look at this concept, and we've worked it up to include it as a sub-study across the dynamic 2, 3 and rectal studies. We got a bit of funding from the St Vincent's Research Endowment Fund to initiate the studies. And it's been discussed with the POCOG, the Psycho-Oncology Research Group, and they've endorsed the study as well. So it's in progress. I'll just go through the concept. So what is fear of cancer recurrence? So there is a definition, a consensus definition of this entity, and it's defined as fear, worry or concern relating to the possibility that cancer will come back or progress. It is an emerging area of research, but mostly the studies have been conducted in patients having conventional cancer treatment. There are validated instruments to measure this concept. So why does it matter, I guess? High fear of cancer recurrence can affect and lead to anxiety and depression, physical symptoms. It can interfere with quality of life, and there is some evidence that it can impact on resource utilisations. And there are interventions that can help lower this fear. Phyllis Buteau from the University of Sydney has an interest in psycho-oncology research, and she actually presented a randomized study of an intervention called Conquer Fear at ASCO a few years ago, showing that this fear of cancer recurrence can be reduced with these kind of interventions. So it's a common feeling, I guess, that patients have. In this meta-analysis across a number of studies, it showed that there is some degree of fear of cancer occurrence in patients in general, in about 70% of cases, moderate to high levels in 50% of cases, and very high levels in about 7% of patients. There is research showing that there are consistent determinants of fear of cancer occurrence, those being young age, patients that have a high symptom burden, and patients receiving chemotherapy and radiotherapy. There is some other evidence which is a little inconsistent about other factors such as female gender, personality factors, having children, objective prognostic factors, but these are less established. There are triggers of fear of cancer occurrence, as you would expect. Follow-up appointments can cause this to be increased, having medical tests, reminders of cancer, and physical symptoms as well. So there we know that there are growing numbers of assays to predict risk of cancer occurrence and treatment response, and those of us or those people that do breast cancer treatment commonly now use things like Oncotype to predict risk and use this to guide treatment. We know very little about the psychological impact of personalized medicine, for example, foregoing standard therapy based on low-risk genomic profiles or high-risk status conferred by the presence of things like circulating DNA and what impact that has psychologically for patients. So turning, I discussed this a bit before, but there are things that can be done for low levels of fear of cancer occurrence, such as education strategies, but for high levels usually requires referral for evidence-based psychological interventions like the one Phyllis Butow has developed. So turning to the fear of cancer occurrence sub-studies, the primary objective then of this suite of studies is to assess and compare fear of cancer occurrence levels in patients participating in Dynamic Rectal, Dynamic 2 and Dynamic 3. The specific objectives are to compare the levels of fear of cancer occurrence across treatment groups at baseline and at the time decisions are made about adjuvant chemotherapy, to determine the prevalence patterns over time in all treatment groups, and to determine potential predictors of fear of cancer occurrence, and also to determine associations between fear of cancer occurrence, anxiety, depression, and health-related quality of life. We've also written into the consent form the opportunity to undertake a qualitative study. So patients are asked to opt in to consent to be contacted at a future time for some in-depth interview work. So we've got that opportunity as well, which I think will be very interesting. So these are the questionnaires. So the Fear of Cancer Occurrence Inventory is a previously validated tool that measures fear of cancer recurrence. It's a 42-item multidimensional measure. There are 7 subscales assessing things like triggers, severity, psychological distress, functional impairments, insight, reassurance, and coping strategies. We've also are using the Hospital Anxiety and Depression Scale and the EORTC Quality of Life Questionnaire. So the way we've set this up is that the baseline surveys are done at the time patients are seen post-operatively for their baseline blood tests. So everyone is sort of on the same level at that point. Time 2 is a point where adjuvant chemotherapy decisions are being made. So those people in the biomarker driven group will know their results. They will either know that they have negative or positive blood tests for circulating DNA. And those in the standard treatment group will also complete the questionnaires at the time when decisions are being made about what chemotherapy they will receive. We're also measuring the same, using the same questionnaires at 9 and 15 months in follow-up, and these assessments at a time when no CT scans are being done that might elevate this particular fear, just in the routine follow-up. In terms of the analysis, we've done a sample size calculation, and that worked out to be a little alarming at 350 odd patients, but I'll show you the progress in a minute. So we're probably about halfway there at the moment. The primary analysis will examine change in fear of cancer occurrence over time and its predictors. The two predictors of primary interest are the treatment groups and whether or not patients are participating or receiving chemotherapy. So our hypotheses are that there will be moderate to high levels of fear of cancer occurrence observed in approximately 40% of patients at baseline, that female, gender, young age, high anxiety at baseline will predict for higher fear of cancer occurrence. And we also hypothesize that patients who know that they definitely have positive ctDNA results will have higher fear of cancer occurrence than patients who do not know their ctDNA result. That is the standard arm of the trial. And they will have higher fear of cancer occurrence than patients who know that they are definitely negative for ctDNA. So in terms of progress in the past 12 months or so, in the Dynamic 2 study, we have about 65 patients with 3 or so time points across 10 sites. So this was added on as a sub-study quite late in the piece for the Dynamic 2 study, but we're happy to have recruited 65 patients across that study. In terms of the dynamic rectal, we have 85 patients enrolled and are collecting their questionnaires at these 3 different time points, and more sites have participated. I think there's about 15 sites participating in this sub-study. And then we've come later to add on the sub-study to the Dynamic 3. So we now have 12 patients who have had baseline and some follow-up, and we're continuing to recruit again across about 8 studies. So that's where we're up to. I would really like to thank Jeannie for allowing the sub-studies to be added to the dynamic suite of trials, and to the study coordinators who are enthusiastically collecting this data pretty much on the smell of an oily rag. The study coordinators at St Vincent's are entering the data into a REDCap database, and we'll continue to recruit. I think my plan ultimately is to combine the data into a meta-analysis type combined study. I don't think the tumour type will impact on the results. So putting all the results together into one analysis, I think will work. I think things like the test results, circulating DNA results, and whether they receive chemotherapy is likely to have more impact on the outcome than the tumour type. So I think that will be a valid thing to do. So we're about approximately halfway in terms of the sample size currently. Thank you very much.
Thanks, Sue-Ann. Maybe I'll just ask one quick question before I go on to the next speaker. Sue-Ann, so let's say we do find that the ctDNA result does affect the fear of cancer recurrence score. How do you see, what will be the next step? How do you see this being helpful in the clinic, and how we can implement this in the clinic? The information from these studies. Yeah.
Yeah, so I suppose if we know that it's a problem and we know there's an intervention that will help, I think identification of it in a screening type of sense will be what ultimately we'd be working towards. But establishing that really is a problem, I suppose, is the first thing. But there really now is a move towards screening for psychosocial distress in our patients. And I guess people that we know are going to go forward to have this kind of information provided at the diagnosis and to determine their treatment, knowing that that could potentially be a problem. I think we would be looking to see and to check that at baseline so that we could intervene over time if that's an ongoing problem. I think it's identifying the problem early so that interventions, some of the interventions now, they're looking, Phyllis Buteau is looking at an online sort of type self-help type intervention. So these interventions may be accessible at a low level fear of cancer occurrence rather than letting it to get to a point where, you know, full-on formal referral is required. So I think that's the kind of direction we'd be heading with it.
And also perhaps if we can do ctDNA during surveillance and they have serial negative results, for example, that may be the next step to look at whether that could allay a lot of the fear recurrence.
Yeah. I think there's a lot of potential for this, and I think it really value adds to the studies to get this kind of information.
Great, thank you very much, Sue-Ann. So let me introduce the next speaker. So we have Associate Professor Justin Yeung, who is a consultant colorectal surgeon at Western Health. Justin will talk to us about his big data research to improve and tailor colorectal cancer treatment.
Thanks, Jeannie. Thanks, Peter, for your invite to talk to everyone tonight. I'm just going to load my screen. Is that good? Okay.
That's good.
Thank you. So all the speakers today have demonstrated our really keen enthusiasm to try and ensure that we get great colorectal cancer outcomes. But what's frustrating for us as clinicians is, why do people get different results? We do know that there's going to be some variations in genotype, cancer type, which determines how they respond to chemotherapy, for example. But at Western Health, our population is diverse. And there must be some other key aspects that we're missing out, that if we can know what's the gap, what's missing, we can then iron out those inequalities. So this slide really represents the three pillars of what we've been trying to do at Western, to try and really iron out those inequalities and try and improve on patient outcomes. We've been focusing on some big data to identify why different patients present at the times they present, whether there's any delays in access, variations of treatment, and ultimately, what effect does it have on their outcomes? We're really passionate about quality of life and trying to introduce PROMS research into clinical practice. And in particular, I'll be talking a little bit about how those from a cultural and linguistic diverse background actually responds to PROMS questionnaires. And finally, I'd like to touch a little bit about some exciting work we've been doing on radiomics to better tailor our treatment for our colorectal cancer patients. So you heard earlier in the session, registry data is king. We also have been very fortunate to be part of the core database, which Peter and others have set up. And specifically, we've also been looking at linking with the CVDL, where we're getting some health and human services data to really work out why we see variations in presentation. So currently, we're looking at presentation across Victoria, in particular colorectal cancers, why some patients present the way they present, why some of them may have delay to a GP, why some of them may be sent for a particular test and not another, and ultimately, how does that affect their treatment outcomes? I mentioned about patient-reported outcome measures, which are key in measuring quality of life, not just due to disease process, but how does treatment affect those outcomes? And there's several different forms of PROMs out there that we can utilize, all very essential. But what we found that is, although we talk about patient-reported outcomes, we're actually focusing most of this on English-speaking population. And if you look at Western, over a third of our population do not have English as a primary language that they utilize. So because of this, we've been doing some really good work with the Peter MacCallum, and thanks to Sandy Harriet and his team, we've been looking into how we can integrate PROMs in the cold population group. And one of the things what we found interesting was that even the definition of English proficiency is quite variable. I just give you 4 examples of patients that I've reached out to doing these studies, and not being able to speak English means different things to them. So one of them said, I can speak English, but not read. Another one says, I can speak, but I prefer to use another language at home, a bit like me. I read at a level of primary school. And there's another one that has said, I was born overseas, I immigrated when I was the year of 1, and now I got a PhD at Melbourne Uni. So all of these may have put down English proficiency, English as a second language, but clearly they all mean different things. And certainly what we need to do is tease out a little bit more about how these are defined and how these affect patients. If you look at what we found after we interviewed patients from a cold background, a cultural and linguistic diverse background, on whether they would be interested in taking part in PROMs research, we actually found integration is probably not as complex as we think. These patients have very similar values, very similar core values, and to them, the answers that they gave meant very similar to what English-speaking patients understood. And I guess rather than focus on how do we actually integrate non-English speaking people into PROMs research, what we should be perhaps focusing more on is the bigger picture. How can we implement PROMs into clinical practice? So I think that will be something that we need to kind of work more on. Finally, I just like to spend a few minutes talking a little bit about a new initiative called AI Precise, where we're using AI technology to measure body composition using CT scans on patients with colorectal cancer to better predict chemotherapy-induced side effects, so that we can ensure that patients have the right treatment with the right dose at the right time. So certainly our goal is to improve our response to chemotherapy, cancer therapy, by personalizing treatments to suit each patient's body makeup. We're all different internally. And we know the importance of being able to work this out because 1 or 2 of us will be diagnosed with cancer. Chemotherapy at the moment is dosed basically on body surface area, which is an estimate based on height and weight. And because it's an estimate, we're going to get a highly variable response. Looking at rectal cancer patients at Western Health for the last 10 years, up to 40% of patients had either a reduction of the chemotherapy or cessation of the chemotherapy due to toxic side effects. And what we don't know is how many of the remaining 2/3 of patients actually were underdosed by the chemotherapy. For those who are not really familiar with what body composition is, it's essentially measuring the key components of the body, mainly muscle and fat distribution, pattern distribution, as well as density and quality. And if you look at this picture here of the Wagyu beef, clearly that's very different to the porterhouse steak on the right. And that difference in meat quantity, the difference in fat distribution, will affect how chemotherapy agents are metabolized and utilized in the body. Currently, to be able to measure body composition is really in the realms of research. The technology is very sluggish, allows you to measure 1 CT slice level at a time using manual assessments, taking up to 45 minutes to measure that. Our technology now allows us to do 3-dimensional top to tail measurements based on CT scans of several hundred slices in a matter of 5 minutes. So 100 times faster, and be able to give us that information and also the pattern of distribution as well. So what's the future of patient-tailored care at Western across Australia? I think listening to all the talks today, I think what we have is a fantastic multidisciplinary group here. We can utilize information on big data to help decide which patients need what treatments potentially, using the body composition information to help tailoring dose and frequency of drugs, using patient-reported outcome measures to determine whether patients should have surgery or should they have chemotherapy, because not all patients get the same quality of life results. I guess what's also I'm interested to working with other partners is getting that genomic data, getting all the biomarkers that we talked about to be able to get this complete package of care. So what are the next steps for us at Western? Obviously, we're keen to get involved with some new prospective trials on using new research therapies to predict dosing. As I mentioned, linkage to genomics. We can certainly expand to other cancer streams. And with this technology, we can also predict complications, in particular bony complications such as osteoporosis and fracture risk. So thank you everyone for your time. I couldn't have done this without everyone's help. And these are just some of the members of the team that have worked really hard with me. Thank you.
Thank you, Justin. It's one wonderful dimension, a different dimension of research you're doing. Perhaps I'll ask the question. I don't think there's any in the chat box. Now, I was particularly interested, obviously being a medical oncologist, about the chemotherapy tolerance and the body composition. As you know, body surface area is extremely crude. Are you planning to do some pharmacokinetic PK collection and data on your project because obviously the pharmacogenomic side of things is also very interesting and might affect how a patient tolerates, particularly including the YPD gene testing. Or is that planned to incorporate that in?
We would love to. I mean, that's really part of our plan, is to reach out to people like yourselves to really work together. I think that's a really important piece of the puzzle. So yeah, love to.
Oh, good. We have some interest in that.
Indeed.
At Peter Mac, so okay. So very fruitful webinar, Peter. Thank you very much. If there is no further question, I will move on to our final speaker of the evening. Thank you, Justin, once again. So our final speaker of the evening is Dr. Koen Degeling. So Koen is a research fellow at the Cancer Health Services Research at the University of Melbourne, and his research is focusing on the use of real-world data from clinical registries, and tonight he will specifically talk to us about how you can use this to facilitate economic simulation model of biomarker-directed therapy. So Koen, take it away.
Thanks, Jeannie, and thanks, Peter, for the invitation to present some of the work that we do within the colorectal cancer space within the Cancer Health Services Research Department of the University of Melbourne, using both real-world data and simulation modeling. So just let me move my Zoom thingy. Yes. So I'll be presenting on behalf of Professor Marta Eisenman, who has been the chair of Cancer Health Services Research since 2018. And he has developed a program of work focusing on research into the economics of cancer genomics, the use of cancer registries in general, as well as improving cancer health services. And today I'll specifically discuss how we use simulation models based on real-world data to estimate the health economic impact of new technologies through 3 examples. So the first 2 examples estimate the cost effectiveness of using biomarker-guided adjuvant chemotherapy for stage 2 colorectal cancer, with the aim to reduce overtreatment in this population and its associated health and economic burden. And both these projects use data from the Australian Comprehensive Cancer Outcomes and Research Database, or ACCORD, that I think everyone will be familiar with, as well as treatment from data from the Treatment of Recurrent and Advanced Colorectal Cancer Registry, or TRAC. So the first project I will present aimed to estimate the health economic impact or the health economic potential, that is, of any hypothetical biomarker to guide the treatment decisions in this setting, based on a sensitivity and specificity for that hypothetical biomarker, as well as estimates on how that biomarker may influence treatment decisions, so whether the results will actually be acted on or not. And this is work that was performed by Rainier Arnolda, who was a Master of Public Health student at the University of Melbourne. So this is the sort of the conceptual structure of the simulation model that Rainier developed. And this model then was able to estimate the health economic outcomes. So if we look at the trajectory, it was specific to individuals who had curative intent surgery for stage 2 colorectal cancer. And then sort of the first decision node in the simulation model was whether the individual would undergo biomarker testing, yes or no. And if not, they would be treated according to standard of care. But if there was biomarker testing, then the sensitivity and specificity would be used to simulate a test result. So to simulate, based on the sensitivity and specificity, whether the individual would get a true positive or true negative test result, or perhaps a false positive or false negative test result. And then the second step was to simulate how that treatment, how that information would then be used. So whether the treatment decision, the treatment recommendation based on the biomarker would actually be acted upon. And then based on the final treatment decision that was made, we simulated the long-term health and economic outcomes. So these are all sort of knobs that we can turn to create alternative biomarker scenarios. So the results from this study were, well, very, very promising. So these figures summarize the outcomes for 100 of different biomarker profiles and scenarios for how these biomarkers may be used in practice. And in these figures, the green suggests, well, scenarios for which the biomarker would be considered cost effective, and the red suggests scenarios for which it would not be the case. And on the left plot, we see that the specificity of the test is considered more important in terms of the cost effectiveness compared to the test sensitivity. And that's because for any sensitivity value, we can see that the specificity will determine whether it's red or green. And interestingly, this suggests that reducing undertreatment is more important than reducing overtreatment. But that's, of course, from a health economic perspective. And if we think about it from an economic perspective, that makes sense because of the high cost of treating recurrent disease. Now, if we look at the right plot, that illustrates the use of the biomarker test, that also has a profound impact on the cost effectiveness. And this plot suggests that it's not only the technical characteristics such as sensitivity and specificity that are important to consider when developing and translating a new technology, and a biomarker test in this case, but also think about how likely it actually is to change treatment decisions in clinical practice. But overall, great potentials for biomarkers to support treatment decisions for stage 2 colorectal cancer. Now, the second project aimed to estimate the health economic potential of ctDNA detection specifically in the stage 2 colorectal cancer population. And this was a study performed by Richard To in collaboration with Jeannie and Peter, which took into account not only the ACCORD and TRAC data, but also data from the phase 2 studies on ctDNA by Jeannie and Peter. So overall, the model structure was quite similar. So there was a standard of care and a ctDNA testing strategy, including for the ctDNA strategy, how likely the treatment decisions were actually to be changed based on the ctDNA test result. But this model was completely tailored to ctDNA based on the observational studies. And in line with the results from the first example, the results for ctDNA specifically also suggested that the use of ctDNA to guide adjuvant chemotherapy for stage 2 colorectal cancer could be cost effective, as it reduced costs for of chemotherapy and increased quality of quality adjusted life years, by reducing both over-treatment and under-treatment. And also in line with the first example, the extensive scenario analysis that Richard performed demonstrated that the use of ctDNA in practice is a very important determinant in the cost effectiveness, and that its predictive value in that, so how likely patients with a ctDNA positive result actually respond to chemotherapy, is very important as well. And that's something that's a quite uncertain sort of evidence piece at this point. So there was quite substantial uncertainty around the point estimates, as you can see by the ellipses here, which is sort of the uncertainty in the evidence, but also the uncertainty in the assumptions regarding the predictive value of ctDNA in this setting. So the results from the randomized studies that are ongoing are eagerly awaited to obtain more certain cost effectiveness estimates. Then the third and final example that I would like to highlight is the simulation work that is done as part of the predicting the population health economic impact of new and current cancer treatments project, or PRIMCAT for short. And PRIMCAT is led by Martin Eisenman, and it's a big collaborative group, and it was funded by the MRFF and started in 2019. And although PRIMCAT considers multiple cancer types, I'll focus on the CRC specific work here, so the work on colorectal cancer. And the aim of this work is to inform the health technology assessment of new cancer drugs for the Australian government by estimating the number of patients that are eligible for treatment at different disease stages and different treatment lines. And that piece of information, so the number of patients that are actually eligible for treatment at a certain phase, is a large uncertainty in the submissions for reimbursement to the Australian government, but also for other governments. So here we develop two versions of our simulation model. Version 1 is the model where we use data from Accord and Track, and that model is almost finished. And then as a next step, we will, let's say, update these models based on a big link data set that the PRIMCAT team is building, which includes data from the Victorian Cancer Registry, Center for Victorian Data Linkage, data on all hospital-related episodes, MBS, PBS, and National Death Index. So what we did here is we created a very detailed simulation model for each different disease stage and disease phase, and we have estimated all the different events and time to events from the real world data. As the treatments for, let's say, stage 3 colon cancer or after a local regional recurrence mainly exists out of one line. There is then potential progression to further treatment, and eventually for example, as displayed here, RAS wild type metastatic disease, which exists up multiple lines. And it goes on after this arrow. And using this simulation, we can look into how each individual will flow through these different parts of the model, and we can zoom into each of these sort of separate treatments and see how often those are utilized. And this is just an example of some of the results we get out of this. We can look at the number of patients who receive any systemic treatment at a particular treatment line, for example, specified by RAS status. And we start by simulating incident cases back in 2000 here, 2010, I believe, all up to, in the future years, so that we also get an estimate of the prevalent patient population. And this is crucial information for decision makers, and it has been unavailable at this level of detail so far. So it's really an exciting project to work on with this great team. And with that, I would like to thank our collaborators on these projects, and specifically the VCCC consumer advisors, a lot of them who are part of the PRIMCAT consumer panel, but especially Sophie Athan and Les Leckie, who have consulted us on these biomarker-related projects. And finally, thank you for your attention. Happy to take any questions.
Thank you, Koen, for a beautiful presentation. Now, without giving away the results of my dynamic study, which is about to report, as Peter mentioned, perhaps next year, what do you think of the role of health economic modelling and well, particularly when it comes to trying to get a test approved by the government or NSAC?
Well, of course, it's an essential part of the final submission phase. If there is to be public reimbursement through the MBS, then the health economic analysis is an essential part there. So definitely at the end is important, but we really advocate to bring health economics as far up front as we can. So as I demonstrated with the first example, we can even do sort of like a headroom analysis or early cost effectiveness analysis based on data from comparators only. So we could, simulate outcomes for a hypothetical biomarker to inform the first trial designs. And then as more data become available, for example, with Richard's study, we can update that. So we can build the evidence, the health economic evidence that we need at the end to get public reimbursement throughout sort of the product life cycle. And by doing that, you can make sure that by the end, you get to the end, you have all the information you need.
Do you think perhaps, I wondered whether it's, we should be talking to the health regulators even before the results comes out or before we start doing the simulation about the type of information they would like to see before we even embark on the project. What do you think? Have you had any experience or know of anyone who's gone through that process?
I know there are some countries who sort of take they try to adopt this product life cycle approach to health technology assessment as well. And especially if we think about biomarkers, we've learned from, for example, Oncotype DX in breast cancer, that's something that people are very interested in, is how likely is the biomarker actually to change treatment decisions? And by engaging with decision makers early on, we can identify those crucial pieces of information to gather those throughout the trials. Because, well, if you get to the end and then you find out that that's something that's very important to them, then it's too late to gather that from the trial. So yeah, that's something that's definitely beneficial.
Good, thank you very much. We don't have any more questions. So what I'll do, thank you, Koen,
Can
again.
I ask a question?
Oh, yes.
You can't see the screen, I suppose. Thank you very much. That was really prompted a lot of thoughts. I'm wondering what the real world experience is in health economics. What's the threshold that usually gets a government over the line when you show a benefit of quite a sophisticated new test like the ctDNA assay? I think what Jenny was implying, I think there's a lot of merit in that. Working out what the threshold would be to get our PBS committee over the line, to know what that is before you go too deeply into a study, because unless they're prepared to change, it's a rather moot point.
Yes, well, the interesting thing is that only the UK has really sort of hard thresholds for how much they are willing to pay for a certain amount of quality adjusted life years gained. And for other countries, there have been sort of ranges estimated or there have been ranges published, but it always depends on the population, the unmet need, etc. And at least for the Netherlands, it's between 20 and 80,000 euros per quality adjusted life year. I'm not sure what the range is for Australia specific, but for NICE in the UK, it's about £20,000 per quality adjusted life year. But for tests and diagnostics, of course, there is this other thing that if it replaces existing tests, then if you demonstrate that it's technically sort of equivalent, but it's cheaper, that may be another argument to sort of bring it to market. So there is no single threshold. And I think the government would be very unlikely to settle on a certain threshold prior to the results being published, because then you exactly know what to work towards in your analyses. To get the dialogue started, to identify the key factors at least that they want to see considered in the analysis would be a great starting point.
Very good. Thank you. Any more question, Rob? Good question, though. All right,
I might make a comment.
Oh,
so,
yes.
So I mean, I think that this is a sort of politics comes into this. So I mean, the reality is, I think that we're very similar to what we have in the UK. That somewhere in the $50,000 to $100,000 per quality adjusted life year gain for drug reimbursement is the sort of range you need to be at. So if you're nowhere near that, you're wasting your time. But the reality is that they factor in what the perceived need is, and there are some patient groups who are much better lobbyists than others. So breast cancer, the threshold tends to be much less. So that's something that, in the colorectal sphere, the patient groups are not that good lobbyists. So if you want, if there's something that we can do, it's to encourage the community to be better at lobbying for their own needs.
Need to find someone famous. Great. I think that brings us to the wrap-up session. I'll hand over to Peter to wrap up the evening.
So thanks, Jeannie. Thanks for all the speakers and the chairs, and a really diverse range of research being presented tonight, which is really what I'm wanting to see come out from very basic research through translational research to phase 3 clinical trials. So a very broad spectrum. Interesting, I think, that all of the speakers presented quite distinctly different types of research. So it's really nice to see the diversity of research, nice to see the collaborations happening within the Institute, particularly that relationship between the clinician and the scientist, which is incredibly valuable. Also starting to see some collaborations across the VCCC sites. I think one of the key things from tonight is hopefully to stimulate awareness of what is happening at the other hospitals and other research institutes, and identify opportunities for collaboration moving forwards. So just to summarise the various presentations. So my presentation is focused on using registry data and combining that with tissue-based research and moving more towards prospective trials and how they could have an impact. We heard from John and Fiona about the work at the Austin, particularly leveraging clinical trials such as the MAX study, multiple publications that have come out of MAX from biomarker-related research, and moving forwards based on some very good science with the Beta clinical trial, repurposing a drug that hopefully has a big impact on patient outcomes. Ian talked us through some of the work that he's been doing, initially starting with administration data, looking at some analysis of different types of surgical approach or different locations and impact, and then moving on to some interesting analysis of clinical data with some very impressive statistical manoeuvring. Rob finished that first session talking about what's happening at a specialised cancer centres such as Peter MacCallum with the patient group that they see, rectal cancer and peritoneal disease. Some of the important work that he's been doing with patient-derived tumour organoids, understanding immune infiltrates, also looking at some diverse types of research which often doesn't get talked about, such as the importance of anaesthesia and the importance of the gas used in laparoscopic surgery. So change of pace after the break and moving on to more supportive care type research. Sue-Ann talked about her focus on patient experience research and importantly highlighted the opportunity for collaboration with Jeannie with the dynamic studies. Justin introduced some more research focused on quality, or more precisely on inequality, and how that may affect different patient groups, delay in presentation, and understanding the impact of culturally and linguistically diverse patient populations. Again, I think demonstrated the importance of data, and that's a bit of a theme coming through, value of linkages across different data sets, including linkages with GP data sets and other data sets that he's been looking at, some interesting work, starting with body composition, how that might predict outcomes, including chemotherapy. Then finishing with Koen, again, talking about a different area, health economics and the value of that. And I think very nicely demonstrating again the potential for collaboration. Koen's been collaborating with Jeannie, looking at the cDNA work with the various colorectal data sets, looking at various health economic modelling using routine real world data. Just to finish again, thanks to everyone that's participated. Thanks for the audience. We're looking forward to some feedback. So there will be a survey that's circulated after the meeting. Very keen to get input into the quality and the content of the presentations from tonight, and particularly opportunities to improve and new areas that could be covered in the future. So please look out for the survey and complete that. And that's it. So again, thanks for your participation and have a good night.